Hi, and welcome to our BioRender 101. Thank you for joining our webinar today. Now, I'm just going to give people a bit more time to join in before we start, so make yourself comfortable and we'll begin shortly. In the meantime, I'd love to invite you to sign into BioRender yourself. If you have yet to check it out, you can sign up for free by visiting biorender.com and clicking on this button found in the top right-hand corner of the site. Feel free to browse through, explore what we have to offer if you haven't already. Feel free to follow along on your own screens throughout the webinar as well. Okay, it looks like most of those who registered are here, so I think we're good to go. Once again, thank you for joining in today and welcome to our BioRender 101. A few housekeeping items before we jump into things today. This session is for you. I'm here to help make it easier for you to create scientific illustrations. With that in mind, please feel free to comment and ask any and all questions throughout the session. The chat is the best place to say hi or chat with others in the audience. But if you have any questions throughout our time together, it's really easy for those to get lost in the chat, especially if there's a lot of activity. So please just pop those questions into the Q&A section in your Zoom window. I want to make sure that you're getting the most out of the time that we have together. With that said, since we'll be together for the next hour, I thought I'd just do a quick intro of myself so you can get to know a bit of this voice that you're listening to. My name is Fuzail Kadri, and I completed my Bachelor of Engineering in Mechanical Engineering at V2 University and went on after that to get a postgrad in Mechanical Design Technology. And today I'm excited to share some of the tools and pre-made graphics that BioRender has for science lovers like you and me. By the end of this session, you'll see how you can so easily create beautiful figures to visually communicate your science efficiently and effectively. But first, I'd like to take a moment to get to know all of you a little bit better. I'm going to launch a quick poll just to see what your field of research is, as well as if you've used BioRender before. I do like to try and tailor my examples throughout the webinar, so your answers to this poll will definitely help me do just that. Right, the poll should be on your screen now. I think we have a few more people to answer, so I'm just going to leave the poll for a few more seconds. Awesome. Looks like most of everybody have answered. And there are some who've used BioRender before, some are just getting started, and there are many who've never used it. So I hope I can give you some tips and tricks today to get you started. And I also wanted to share a link in the chat for everyone who needs access, which is premium BioRender access. So this is a form that you would need to fill. to receive access. And once you fill up the form and send it over, someone should get back to you and then help you with receiving premium BioRender access. And that's the link in the chat. Awesome. So let's just dive right in. A big part of the BioRender team used to work in research, and we know that communicating science is hard. Scientists spend over 1 billion hours working on their visuals, which slows down the research process and wastes countless dollars on errors and delays. We're giving scientists time back by supplying them the tools and practical knowledge that they need to visually communicate their research. The functions that we're reviewing today are applicable to a wide range of use cases. Presentations, grant figures, publications, graphical abstracts, and posters are just a few. Really, you should consider adding a visual anytime you find yourself explaining what something looks like in your writing. We also run webinars that cover graphic design tips for specific types of figures like grant applications, graphical abstracts, and pathway illustrations. Keep an eye out for more information. But let's get down to business now. So to start, I'll show you how to use our icons and brushes. Then I'll demo how you can create figures using our pre-made templates, as well as how to create a figure from scratch. Once our figures are complete, we'll review how you can export your figures. Next, we'll go through the share and collab features that we have in BioRender, which make it really easy for you to work with your colleagues. Finally, we'll wrap up today's session with an overview of our other main features. This includes our PowerPoint integration, graphing, and poster builder. Are there any questions before we jump into the basics? Awesome. So I was actually presenting in BioRender itself. And we do have slides, which is a premium feature and which is available for you to use to create your presentations right in BioRender itself. But let me back up a bit because I know that some people here are new to BioRender. So when you first sign into your BioRender account, this is what you'll see. You have your gallery on the right here with the illustrations you've created. And on the left, you have the ability to organize and group your illustrations into folders. At the top are all the folders and figures that I've created myself. They are private to only me unless I share them. At the bottom, as you can see, are all the folders and figures that people have shared with me. We'll talk more about this sharing feature later on. First, let's create an illustration. And to do that, I'm going to click Create New and then choose Illustration. And this brings me to a blank canvas. And just like PowerPoint, you can change the canvas size and color. You can also go over here to add more slides. Let's say if you're creating a presentation or making multiple figures. Now, since I've already made slides for today, I'm just going to switch over to those. Now, what makes BioRender such a time saver are our icons and brushes. We have over 55,000 icons and all of them are vector-based, just like this one over here. So you can make them as big or as small as you want and they won't become pixelated. To find an icon, simply go to the left-hand side, click Icons, and you'll be presented with a list of all the different categories of icons that we have. Definitely explore around here to see what you can use in your work. You can also use the search bar at the top to look up any icons you'd like. Awesome. For my example, I'm going to switch to the next slide and I'm going to start by searching for an antibody. Let's choose this one. I'm going to drag that onto my canvas. Now for any icon, you have a few customization options. You can use the blue borders surrounding the icon to resize it and to rotate it as necessary. As you can see to the left-hand side, you can change the color of the icon. Our color presets are here to make picking icon colors easier for you. However, if you're looking for, let's say, a very specific type of color, you can explore options around here or go to the color overlay option and then click this plus sign, choose between the wide range of colors available, or just enter an RGB or hex code. Some of our icons have layers. Let me show you a bottle, for instance. Now, at the top of the sidebar, you can see that this bottle has three layers: bottle, liquid, and cap. If I click on any one of those layers, I can change the color of that specific part of the icon. Let's change the cap to, let's say, this color and the liquid to this. And perhaps that then matches really well with what I actually have in the lab. Now, all the icons I've shown you so far are regular icons. While there are colors and layers that you can play around with, they're essentially a single unit. So you can change it as a whole, but you can't change anything within it. So our second group of icons, however, are much more customizable, and these are called grouped icons. And you can identify them by this purple box around the icon that you see here. Now, these are essentially individual icons that are clumped together. Let's take a group of stem cells, for instance. So when you click into it, you can actually edit each of the individual icons any way you want. So I can delete icons or I can rearrange them. Let's say that one of these cells has a mutation. So I can change its color and even add a glow if I perhaps visually tag this mutated cell. Transparency is also a really cool effect that you can take advantage of, as making certain icons more transparent gives them the illusion that they are further away, as you can see with these more transparent cells in the background. When I click out of it, I don't have to worry. It snaps back into a single grouped icon that can be moved and can be resized altogether. You can also add to and remove icons within groups. For instance, I'm going to create a mouse with a tumor. Let me search for a mouse. Then I'm going to add a few tumors. You can see that all our icons are meant to work well with each other. Everything is made to look like one artist did it all. Now, to group these icons together, I can just select all the icons and click on this group button on the top on the toolbar. And now, as you can see that this is a grouped icon indicated by this purple box around it. And this way I can resize it as a single icon and move it all together. Now, if you feel like you'll need your grouped icon again later on, you can add it to your favorites by selecting the icon and clicking this favorite button on the top on the toolbar. This will bookmark it in your favorites tab found on the left sidebar so that you don't have to create it every single time you need it. You can just grab it from here. Now, I know that this mouse with a tumor only took me a couple of seconds to make, but when you have grouped icons that are much more complex, saving them to favorites this way will save you quite a bit of time in the end. Another great benefit to favoriting icons is that these favorites can also be shared with the team members to standardize the way you all visually communicate and make all communication more uniform and consistent across the team. The next type of building block you'll want to take advantage of are our bio brushes, which you can find through your search on the left here under brushes. As you can see, there's a variety to choose from. We have phospholipid bilayers, DNA strands, membrane brushes. For my example, I'm going to show you this blood-brain barrier brush. Compared to icons, brushes are much more dynamic in nature. As you can see, they have both blue and white nodes. These allow you to manipulate the structure of the brush. If I click and drag a blue node, it will automatically add more subunits to lengthen the membrane. Blue nodes are also inflection points. So by clicking and dragging a blue node, I can change the shape of my brush. White nodes are places where I can create new inflection points. After you click a white node, it turns blue, and then I can contort the brush and create this smooth inflection point. See how quickly I was able to create that membrane. I saved so much time not having to copy and paste and position each individual component myself. And just like icons, I can change the color and I can add a few effects to the brush. Hopefully you have a good grasp of the building blocks you can use in BioRender to create your figures. Does anyone have any questions on what we've covered so far? Awesome. So let's move along to our next agenda item, which is figure creation beginning with templates. And I'm going to move along to my next blank slide. You can find all available templates by clicking templates to the left over here. This will give you a breakdown of all the different categories that we have. If you don't see a specific category you're looking for, you can always use the search bar at the top. Templates can be really useful if you need to introduce some background information on protocols during a meeting before discussing the findings of your current work. Let's say that I work with tissue samples in my lab. So I can search for a tissue preparation template. I'll be presented with a variety of options. Let's say this tissue sample preparation template looks like a good one for me to use to showcase a lab protocol. I can click on each template like I did to learn more about it. And then if I like it, I can just replace it to my canvas or just add it. All of our templates are essentially just large grouped icons. If I double click into this template, you can see that it's just made from pieces and icons that are readily available to me on BioRender, which means that each template is entirely editable. I can move things around, delete unnecessary parts, add what I like. Everything is customizable. Let's say that I don't use liver tissue in my lab. I use heart tissue. So I can search for a heart to replace the liver in the template. But first, I'm just going to delete this liver. I'm going to go back to icons and then search for a heart. And let's say it's this one that I will be choosing. Awesome. I'm just going to bring this scroll line to the front. And to do that, I'm going to click and select that icon, then right click it, and then go to arrange and then bring it to the front. Let's say my tissue dissociator is also of a different color. So I'm just going to choose one from these color presets. And there we go, a protocol slide that I could quickly and easily add to my presentation. If you take a closer look at all the templates that we have, there are also less fleshed out templates that are available to you. And I would encourage you to search for these and use them as a more general layout to start your figure with. That way, you don't have to worry about arranging everything together. You just need to slot things into the framework provided, which will hopefully further accelerate your figure making process. Next on our list, we're going to create a figure from scratch. Let's use a DNA example. So I'm going to start by searching for a chromosome. And there's a lot of different options. Let's choose the simple centric chromosome. Next, let's add in some DNA. I'm looking for something tapered, so it looks like it's coming out of my chromosome. So I'm gonna just type DNA and write the word tapered. This one looks good. Bring it onto my canvas, enlarge the icon, and just place it onto the chromosome. That's perfect. Next, let's extend the DNA by grouping it with another DNA icon. So I'm going to go back and search for, let's say, DNA and then use the short DNA icon to complete my figure. Now you can see that there is a slight misalignment. So I'm going to increase the size of the icon. And let's say if I'm not able to fit it exactly using my mouse, I can just use the arrow keys and then place it into position. Awesome. Now I can change their colors so they can be more cohesive with the blue chromosome. To do that, I'm just going to select the icon, go to the preset, choose this bluish color, and I will then go ahead and group these icons together. So I'm just going to select them both and hit group. That looks good. Now I want to make the viewer feel like they're zoomed into a nucleobase level. And to accomplish this, I'm going to search for a callout. These are my favorite for any zoomed in effect. This one here has a square bottom. Let's go with that so we can still see the DNA. I'm going to rotate it and just place it, let's say, on top of this part of the DNA. Now I'm going to double click into my callout group icon and I'm going to extend this top square into a rectangle so I can place in my nucleobases. And I'm also going to realign the shape at the bottom so my illustration looks seamless. Awesome. Next, I'm going to scroll through my DNA search again and pull out these nucleobases. There they are. So I'm just going to enlarge them so they can fit perfectly in this rectangular box. And now to make the best use of my space, I'm just going to double click into this icon and then delete the parts, some of the pairings, or an easier way for me is to just select that icon and use a crop feature from the toolbar. I'm going to use rectangle crop. I'm going to change this section and then double click to crop it. And there we go. I was able to quickly crop and place the nucleobase inside the callout. And all I have left to do now is add in some text. So I can add a title by using this text function. I'm going to call this title. And I'm also going to add a subtitle. But before that, I will select the font and make it bigger. Let's say I'm going to make it to size 20. And I'm also going to change the color. So let's make this all capital. And let's change the color to the color of the chromosome, for which I'm going to use this color picker option, which makes it really easy to pick the color of my choice. And now the title looks cohesive. Next, I'm going to type in the subtitle, for which I'm going to duplicate the icon I created. And to do that, I'm just going to select the icon and hold the Option key on my MacBook, left click and drag it to duplicate. And I'm going to name this a subtitle. Perfect. Now I just need labels for my sugar phosphate backbone and my nucleobases. So I'm just going to go to the text and I'm going to go ahead and type that in. I'm going to duplicate this and call this nucleobase. And I'm just going to reduce the size. I'm just going to make this 10 and I'll do the same for the other. I just want this in a straight line, so I'm going to extend the text box. There we go. Awesome. Now I just need labels to complete the labeling, and you can use this lines feature for drawing those labels. And I recommend using the lines with the dots at the end, and they make it really obvious that this is the terminal point that you're identifying. So for the nucleobases, let's choose this, and I'm just going to use the option key to again duplicate it for the sugar phosphate backbone. I'm going to reduce the size and rearrange my labeling. I'll do the same for the other label. And there we go, a figure made from scratch in less than a few minutes. And once we've made the figure in BioRender, let's talk about what happens next. So once the figure is done, you can export it by using this export feature in the top right over here. You can choose to export either all or select slides. I'm going to export slide number 14 because this is the figure that we created from scratch. You can also choose to export your work as PNG, JPEG, or a PDF. And with your premium license, you'll be able to export in higher resolutions as well. If you're planning to publish your illustration in a manuscript, then make sure you hit the publication permissions button at the bottom, and it is proof that you own your visuals. When I click export, you'll see that this file is saved directly onto my computer. I'd also like to bring your attention to the citation prompt that may come up. So make sure you're writing created with biorender.com in your publications, presentations, and anywhere else you're sharing your illustrations. For me, it showed up if I'm looking for a publication license because I checked the box. Let's say I am. So I'm going to click Get Publication License. It will ask me which slide. Let's say it's the one we made from scratch. And where am I submitting this to? Let's say it's for a journal. I can type the name of the publisher. Manuscript number is optional. Just have to select if this is an open access journal or not, and then download the publication license. If you'd like to collaborate with other team members or get feedback on your completed work, you have a number of different options. First and foremost, you can also share your illustration by clicking up here on the share button and then entering your collaborators' emails. BioRender saves your work in real time, so everyone can collaborate on the same illustration at once. You can even add comments on the canvas itself, so you don't have to email your team back and forth. Apart from just sharing single illustrations, you can also share entire folders. This is super useful, for instance, when you'd like to have a single database where all your live files are created. Let's start with groups. To do that, let's click on your profile in the top right-hand corner and then choose Edit Profile and Settings. This will bring you onto your profile, and on the left-hand side under General, you can click on Groups. Here you can create a new group and then add your team via email. Let's navigate back to our gallery page. Now I can create a folder, for example, like this one, and I can then share the entire folder with my team. Or I can even do it individually via email. You can also share favorites with your group. So let's click back into our presentation. Let's say if there's a combination of icons that you've created and you would like others in your lab to reuse, then I recommend grouping them and then favoriting them. I can do that with the chromosome and the DNA here. So I'm just going to select them both. And I'm going to hit the Command G shortcut on my MacBook to group them. And then I'm just going to click the favorite button to add them to the favorites. So I can use my favorited icons on all my file types, and they won't change even if I do modify the icon on my canvas. So I can also click on these three dots beside the icon, the top right-hand corner in the favorites tab, and then share the icon with my group as well. Comments are another collaboration feature you can use. Let's say that I think the color of this chromosome should be different. I can just click to put a comment right next to it. So I'm going to choose comment from the toolbar, and then I can type in my suggestion. If I'd like, I can even tag team members directly and they can receive a notification. Then my team member can click into the comment, reply to it, and then resolve it. I also wanted to show you all the shortcuts that you can use. Since we used one for grouping, they can be found under the Help and Keyboard Shortcuts section. And the most relevant shortcuts based on if you're using a Windows or a MacBook will be available. Now that's all for collaborating. Does anyone have any questions before we move beyond the basics? Awesome. So BioRender has three other big features that can help you speed up and improve the quality of your scientific illustration. We don't have time for an in-depth review of each today, but I'm going to give you a high-level overview, and you can learn more about it at our next training, which is the BioRender 201 for new and advanced features. Let's start with a preview of our Microsoft PowerPoint integration. To do that, let's hop back into the slide we created from scratch. I'm going to drop some links to the Microsoft App Store in the chat. Once you follow setup instructions, you can log in into BioRender within Microsoft PowerPoint and then perform the same actions that I'm going to show you. In my case, I'm just going to begin by clicking on Copy to PowerPoint button here on the toolbar. And I'm just going to go to a PowerPoint blank slide. I'm just going to paste that up here. And there we go. It's that easy to bring your illustrations into PowerPoint. But the more exciting part of this export option is that the figures are still synced to your gallery. So let's say if you modify your original image, then you can simply hit this update image button to save you from having to export and reformat your illustration again. Let's go ahead and try that. So I'm just going to go to my um, presentation again. Let's say I'm changing the color of this chromosome to, let's say, yellow. Now, this is an update I made. So if I go back to my PowerPoint presentation, it will show me an update was found on so-and-so date and time. When I click update image, then that should now reflect here in the presentation. If you're interested in installing the PowerPoint add-on, then you can find it here, just this link that I'm pasting in the chat. Let's say if you're having issues with installing the add-on, you might need to contact your IT team so they can help with the process, because sometimes that could be blocked. But yes, let us know if you do face any issues. They'll be happy to help. Now, if I go back to my presentation, now we can take a look at our next option, which is the graphing in BioRender. So from our gallery page, we can click Create New, and then we can choose Graph. We have four different options for datasets that include differences between groups, linear regressions, and dose responses. One neat thing about BioRender is that we surface the compatible analysis, which makes it really easy to choose. We also show visuals of what your end result could or will look like. In our example, let's go with differences between groups with one independent variable. By default, BioRender provides demo data to help you get started. You can easily delete this data and then just paste in your own data. As you can see, I have a control group and then three groups with corresponding results data. This data is stored in my dataset. Automatically, BioRender has created a column graph to plot the results of these groups. I want to demonstrate whether or not there is a significant difference between them. If I wanted to, I could show this plotting as a column box plot, let's say a violin plot, which is a new feature, or a mean median point, mean median line. Now, let's click column, let's click violin actually for our example. Now, to analyze these results, I need to conduct a one-way ANOVA analysis, which is very simple. I can just click on Run Analysis, or I can just go here and say Add Analysis. My groups are pre-selected, and I can see my analysis will have a significance level of 5. I'm going to click Continue because I want to know which specific pairs of treatments are significantly different from each other. I'm going to use Tukey's multiple comparison test. So in this case, first, I'm going to keep the experimental design as default, the test type also as parametric and default. And if there are variances of standard deviation, then I'm going to choose and say yes. The only thing I'm going to change is if I'm running multiple comparison tests. So I'm going to click that and say, let's say sample group means are compared with each other. And if there's a test correction method, the app suggests the best one for me, and I'm going to choose Tukey in this case. Of course, if you ran a different comparison test, you will have different comparison test correction methods to choose from. But now we have a one-way ANOVA with Tukey comparison test. So I'm just going to hit run. And BioRender then provides me with a full analytic report, calling out that some of these treatments are different from others. I can dig into the details of this report if I want to review descriptive statistics. However, I want to not have my audience read through this report. So I want to communicate these findings on a graph. So I'm going to go back to the graph and I'm going to enable this option for data points. I'm also going to show all the other groups that were compared. And these were ignored because they had non-significant values. I can just click on that and then change these asterisk values to p-values. Now, because I built this graph in BioRender, I can easily add it to an illustration alongside other figures and then share it with a colleague to get feedback. Before that, I can even change the appearance, like I can change, let's say, the color scheme. I can even change the font and how the data has been displayed. But in our example, let's go ahead and give this graph a name. I'm going to call this graph 1. And I'm going to go back to our illustration. And let's say I want to add this graph to the figure I created from scratch. Now, you can do that by using this graph option on the left on the sidebar here. And I can just select my graph and incorporate it into the figure. What else I can do is resize the graph and the scale will adjust to the new proportions and maintain its accuracy. Don't forget that you can also bring this graph into your PowerPoint presentation. So I'm going to try to place this graph to the right over here. And I'm going to go ahead and change the canvas size. So I'm just going to zoom out a little bit and then use this section to expand it and apply it. And there we go. In a few minutes, I use simple graphing presets to run a common analysis, visualize the results, and standardize the look and feel of my science communication in BioRender. There's more that can be done in graphing, but in the interest of time, we're going to move along to a next feature, which is the poster builder. But before that, I see that there's a question from Kimberly. Thanks for your question. It says, so BioRender performs statistical analysis. What is the limit on the data set size? That's a great question. So if I go back to the graphs, and let's say I hit create new, so there's definitely a limit since this is still in beta and our team's building it. This is the first iteration of the graphing. So the data set does allow you to paste to a certain limit of data, but for now it shows 50 rows. Yep, but I think it just keeps increasing as I keep pasting more data. So I believe there isn't a limit or so as of now, but I can verify this with our team and then get in touch with you. I hope that helps answer your question. One more thing I wanted to show is there are different types of formats that you can paste your data into. Now, this is a long format. There's also a wide format if you need more columns, for instance. Awesome. So last but not the least, let's create a new poster using the poster builder in BioRender. And to do that, I'm going to go back to my gallery, hit create new, and then choose the poster builder option. Now, since I have a BioRender company account, I can see all the templates that are available to everyone. However, you'll only see the basic templates and the custom templates that are available to your organization. You can filter the templates available to you by poster orientation by clicking on this option on the left. Let's say that I want to create a poster with a landscape orientation today, and I'm just going to search, let's say, for a classic template. Let's say this one looks good, and I'll click and then use this template. Great. I've got my template loaded. And on the left-hand side, you'll find the primary toolbar that you'll use to build your poster. If I click into any part of my poster, this is where I'll be able to change its design. Let's say like the font or the color. And when I click out of my poster, this is also where I'll be able to drag and drop different content blocks directly onto my poster. In our poster builder, you need to create your layout before you add in your content. Let's say that I want to add an abstract section right above my introduction. So I can simply drag and drop a section on top of my introduction. You can see that there are these blue lines that show up telling me where my new section is going to be placed. For instance, this tells me that my new section would be placed on top of all my columns, like so. And that's not what I wanted. So I just wanted to drop it on top of my introduction. So I'm going to undo this and then drag the section. And now the blue line shows correctly. And there we go. Now I'm just going to double click into the title and call this abstract. I can just as easily delete sections. Let's say that I don't need this method section in my poster. So I'm just going to click on it. And then I'm going to click this trash button to delete it. I can also drag additional content blocks into any section I'd like. Let's say that I would like to include a figure with my abstract. So I'm just going to go ahead and choose this image content block and drop it here. And as you can see that when I add any image blocks to my poster, BioRender automatically adds a caption block under the figure for me. All right, time to add some images to my poster. So when you click on Add Image, in the middle of the image blocks, you'll see your BioRender gallery and all the files that you've created. You can also add images that you have outside of BioRender by clicking on Add Image and then choosing this Upload section up here. Here you can upload a file. For example, I have this adipose tissue image that I'll be adding to my poster. Now note that when you resize one section, the other sections will auto adjust as necessary. So you don't have to take the time to resize everything manually. If you go to the top toolbar here, you have some other poster settings that you can play around with. You can, for instance, change the size of your section margins. There's just a space in between each of your sections. Let's say if you need a bit more space for your content. Now it's 0.75. I'm going to make it 1. You can also change the size of your content margins, which essentially is the padding that you have for each content in the section. I definitely recommend taking some time to play around in the poster builder. Having said that, we've actually made it to the end of our wrapping beyond the basics and everything on our agenda. If you're interested in more in-depth information and tips for using BioRender, then please consider attending our other sessions. A BioRender 201 for new and advanced features recaps what we went over today and includes in-depth walkthrough of our poster builder, graphing, and PowerPoint integration. We also demonstrate our protein data bank integration. And after that, a BioRender 301, which dives into the details, which expands on how you can fine-tune your illustrations. We review different icon color, transformation, and crop options, alignment, and everything you need to know about circular lines. Right now, I'm just going to post a survey link in our chat. Please take a couple of minutes to fill out the survey. So we truly do want to continually improve. And so your responses to this webinar feedback survey will really help us better support you and everyone else in the future. But that's about all I have for today. Hopefully you found this webinar useful and informative. And once again, if you're interested in getting a premium license with the department, then please fill out the form that I've pasted the links to in the most recent message in the chat, and you'll then be directed to receiving premium access. Let's say if you need more help in working with BioRender, then you can always click on this help button at the bottom right-hand corner, and you can just type in anything that you need to search. You can also click here to get connected with a customer experience and support team, and they are amazing and very timely with their responses. So I'm also going to stick around for a few minutes. If you do have any other questions or curiosities about BioRender, I'm more than happy to help. If not, I hope you have a great rest of the day. Have fun with BioRender yourselves. Thank you once again for joining this session, and also have a wonderful weekend. We have a question. Will this presentation will be available to us after it ends? So I'll be sending a recording to everybody who attended the webinar, and also those who registered. And the presentation will be a part of the recording, but it won't be sent to everybody personally as a separate file. I hope that helps answer your question. Very welcome. Any other questions before we wrap up for today? Awesome. I see there are no more questions. So have yourself a wonderful rest of your day, and take care.